whoever you are, from the serendipitously cisgendered to everyone else. Hello. Today's topic was foreshadowed way back in episode 2 when I said something like, quote, "We must be especially skeptical of labelling infinitely graded yet politically sensitive attributes like mental health, ethnicity, and gender." End quote. Well, today the rubber hits the road. The last few episodes of Wiggle Soup concerning genetics, origin of life research, and quantum field theory have all had one thing in common. They are all horrifically complex topics. And everybody knows they're complex topics that communicators and popularizers bend over backwards to explain simply. This episode is the opposite. I plan to take something that most people assume is very simple and explain why it's actually very, very not simple at all. Last episode, I mentioned that an organism's sex is usually defined by the size of its gametes. That is, the reproductive cells that it produces. Eggs, which are relatively large compared to sperm, or sperm, which are relatively small compared to eggs. But I had to tactically ignore that many species don't have two distinct gamete sizes. Some microscopic organisms actually have two sexes, or mating types, but the sperm and eggs that they produce are the same size. And some creatures, like soldier termites, are sterile, never making any reproductive cells at all. And while we're limbering up, did you know that there are some species of fruit fly who produce egg cells of one size but have 3 different sizes of sperm, one of which is bigger than the egg itself? Does this mean that species of fruit fly has 4 sexes? Spoiler, there are no easy answers in reproductive biology. Anyone who claims otherwise is probably talking bollocks. And there's a lot more to grasp than that. So before these idioms get any more awkward, let's take a dive into all the biological nuance and neurological variables that expose sex and gender as anything but simple binary categories. At first, it may seem convenient that you belong to an anisogamous species, with 2 distinct sizes of reproductive cells. But the human way is not the only way. Other earthlings like oak trees, leeches, and cucumbers also possess distinct sperm and egg cells, but they make do with only one sex, and everybody produces both. Such organisms are called monoecious if they're plants, or hermaphrodites if they're animals. Most backboned animals, including humans, typically have 2 normative sexual presentations that correspond with these 2 types of sex cells. We are, therefore, a dioecious species. But the fact that most individuals fit into one of these 2 main categories does not mean that only those 2 categories exist. To take some non-contentious examples, traffic usually peaks twice a day at rush hours, but there are obviously cars on the road at other times as well. If you're looking for someone, the 2 most obvious places to check are where they live and where they work, but they could also be somewhere else. And restaurants do most business over lunch and dinner, but they do serve customers at other times too. Stated scientifically, we call this a bimodal distribution, where most data points cluster into 2 dominant groups. However, a bimodal distribution is not the same as a binary distinction. Sex, in humans, has a bimodal distribution, but it is definitely not a binary distinction, as I hope will become increasingly obvious. Amongst dioecious creatures, many are also sexually dimorphic. This means that typical male and female characteristics appear visibly distinct. However, there are animals with two sexes that appear almost indistinguishable. Hyenas are a prime example, as female hyenas even sport a convincing pseudopenis. Despite hyenas, swans, squirrels, and other monomorphic creatures, sexual dimorphism is one of the most common kinds of diversity found in nature. Humans are a classically sexual dimorphic species, but these differences can confuse what it means to be male or female. To illustrate this, how many anatomical features could an animal get rid of without forsaking its sex? Little baby barnacles from a group of crustaceans called the rhizocephala are released from their mother into the ocean and swim about in the open water for a few days. They don't eat as larvae as they have no mouth or digestive system. Their only goal is to locate and settle into the exposed limb joints of unlucky crabs. Having securely attached itself to this most vulnerable bit of a crab, a young female barnacle injects a mass of cells from its syringe-like head into the host's body. These cells grow into long branching threads that extend right through the crab's soft tissues, tapping into the host's organs for nourishment. Being a female, this diffuse, shapeless, but singular parasite then grows an external ovary lump, which emerges from the crab's abdomen. The ordeal is an understandable burden on the crab, who is effectively sterilized but left without any other physical impairments. What about the male rhizocephalan larvae? Well, these dudes, attracted by pheromones released by an established female barnacle, seek out and inject their cluster of cells directly into that sexy, exposed, parasitic ovary lump. These male cells also grow like a fungus, but through the female's body, which is mostly just one massive ovary. Here, male tendrils set up shop as a sperm-producing factory, spending the rest of their existence directly fertilizing new barnacle eggs as the ovary produces them. If a male rhizocephalan reaches this advanced stage, it can be considered winning at life. Although, as multiple males can all burrow their way into a female gonad at the same time, paternity isn't completely assured. Still, there's only so much a guy can do. Being nothing more than a parasitic sperm-building cell network within a parasitic egg-building female, herself within the body of a still free-living crab, adult rhizocephalans probably represent the most basic form of male or female that can exist in the animal kingdom. As if we need reminding, they show us that the difference a creature's sex makes to its life trajectory can be enormous. Being born male or female influences not just how you look, but how you act, your internal anatomy, lifespan, resource requirements, and even the sort of diseases you can catch. And all of these factors influence the reproductive strategies of different mating types within a species. If you produce both sperm and eggs, like an earthworm, sexual encounters become a battle to fertilize a lover without letting your own eggs get fertilized back. Winners are those with all the babies and none of the responsibility. Other creatures, like some fish, are sequentially hermaphroditic, becoming male and female at different times in their life cycle. Now you might be thinking, why get so bogged down with all of this? Surely we can just use X and Y chromosomes to determine what sex a creature is. That would be great if all animals had different sex chromosomes. Using chromosomes to steer the sex of your offspring is the norm for some reptiles, some fish, a few arachnids, most flies, all beetles, and yes, us mammals. And with mammals, all embryos start development as females, including humans. As we covered last episode, chromosomes are large bundles of DNA that carry lots and lots of genes. In humans, all our chromosomes, other than the X and Y chromosomes, are collectively called autosomes, and these come in matching pairs. When making sperm or eggs, autosome pairs splice together and exchange chunks of genetic material in a process called, wait for it, recombination. Generally speaking, recombination keeps an autosome pair looking pretty much the same across successive generations. Both chromosome number 3s, for example, look very similar to each other, but different to both the chromosome number 9s. For unknown reasons, at some point in mammalian evolution, one pair of chromosomes, the so-called sex chromosomes, stopped recombining. Eons of divergence has resulted in the mismatched X and Y chromosomes we behold today. This gradual change can be traced back through the relationship history of all mammals. From monotremes to marsupials to us placentals, recombination became outlawed across larger and larger portions of the sex chromosomes. In species where the presence, or more accurately the dose of X and Y chromosomes determines the sex of the offspring, XX is usually associated with female traits, while XY spits out something more male-aligned. Notice the mismatched chromosomes results in a male. Birds do something similar with W and Z chromosomes, but females get the mismatching WZ pair. Oh, and all bird embryos begin development as males. Coincidence? Probably not. In bees, wasps, and ants, there's no chromosome mismatching going on. Females will develop when an egg is fertilized, and there are 2 matching copies of every chromosome. Unfertilized eggs, with only 1 set of each chromosome, will develop into males. Here, where there's no pairing up of chromosomes, males receive no genetic material from any biological father. They are all mummy's boys. Because of this genetic quirk, sister bees, wasps, or ants will be more genetically related to each other than they would be to their own offspring. The quirk of having a greater genetic interest in the success of the colony over making your own babies inspires many altruistic and self-sacrificing behaviours in these insect sisterhoods. It is true, though, that possessing sex-specific chromosomes is the most common way of determining an individual's sex in the animal kingdom, though lots of reptiles and fish use environmental triggers instead. In many cases, it's the temperature of the eggs while the embryo develops that makes the difference. Subjecting turtle, snake, and crocodile eggs to different temperatures will change the sex of the hatchlings. Cool temperatures produces females. Warm gives you males. But very warm spawns aggressive, hot females. Nothing is simple. And the mechanism raises troubling conservation issues, as global warming may skew sex ratios in future generations of these creatures. A warming climate may stop the production of males entirely. Another natural environmental trigger is the absence of one sex within a community. Sex changing is no big deal for hundreds of species of fish, reptiles, birds, amphibians, and invertebrates. Clearly, one way to improve your chances of being a parent lies in being able to assess your social surroundings and swap sex to meet demand. For many animals, being a large male can yield reproductive benefits, especially where males have evolved to fight over females. In other species, there's an advantage to being a large female, as greater size means you can gather more resources and lay more eggs. In either case, the death of a dominant individual of the opposite sex may present an opportunity. Events like this can trigger a sex change in the next largest individual who steps in to claim the spoils. often suspiciously fast. But let's get back to humans. We're simpler than all that. No haplodiploidy for us, thank you very much. For us humans, it is all about sex chromosomes. But there is still no universal XX versus XY dichotomy. Some humans are born with 3 sex chromosomes. Here, XXY is called Klinefelter's syndrome, and XYY Jacob's syndrome. Some people have 1 solo X chromosome, which we call Turner's syndrome. Or you could have 3 X's, triple X syndrome, or 4 sex chromosomes, XXYY, which is imaginatively called 48XXYY syndrome. So sit back down, we're going to need a more nuanced view. What is it that makes our sex chromosomes so important? The human X chromosome carries about 800 different genes on it, and it's really not that different to any of the autosomes. The X chromosome, in stark contrast, only carries about 70 genes, and less than half of those are unique. However, despite the impoverished and diminutive nature of the Y chromosome, it does hold a few genes vitally important to the building of gonads, both testes and ovaries. You see, even with a normal, healthy 46 chromosomes, including a textbook XX or XY, the idea that chromosomes determine what gonads you have, and that gonads determine your sex, is still problematic. This is because every individual mammal possesses the genes for building both testes and ovaries in their DNA. Getting one or the other, or neither, or both sets of gonads comes down to several cascades of interactions between the genes on several different chromosomes. As we get into this, feel free to Google as we go. There's a gene on the Y chromosome called SRY, which is important in the development of testes, but only in the most insanely convoluted way. Like having the central heating and air conditioning continually working against each other, female bias genes on other chromosomes keep pushing gonad development towards ovaries. The gene that actually builds testes is called SOX9 and is found on chromosome 17, which everyone has. Now, hold tight. The gene NROB1 on the X chromosome makes the protein DAX1, which stops SOX9 from building testes. But males have an X chromosome too. So the SRY gene on the Y chromosome makes a protein that sticks to NROB1, stopping it from making DAX1 so that SOX9 can build testes. This is difficult without a diagram, but it's just the start of how complicated sex determination is in human beings. And if you haven't guessed, every single one of these genes, steps, proteins, and inhibitors that I just described is subject to interference. Any chance duplication or upregulation of the DAX1 gene can overpower SRY's dampening influence. Such individuals can develop ovaries even though they possess a Y chromosome. You could be quote-unquote male with a healthy Y chromosome but a faulty SOX9 gene on chromosome 17, and so develop ovaries. Or you may be genetically female with no Y chromosome but a faulty NROB1 gene, granting you testicles. To poke at this mess, when spliced into embryonic female mice, the SRY gene causes some, but not all subjects to develop testes, penis, and male behaviours. Cutting SRY out of males leads some, but not all lab rats to develop female reproductive organs and female-associated behaviours. Some of those XY male mice can even give birth to fertile offspring after getting pregnant. Back to humans. Another gene called DMRT1 also plays an important role in the development of testes. This gene stays active after gonad development is complete. If the effect of this gene is removed at any point in a male's life, reproductive cells will revert back into ovary cells, effectively changing the sex of the individual. For similar reasons, in female-presenting individuals, outspoken male-biased genes like DMRT1 must be continually silenced. Sex determination is not a question of yes or no, on or off. It's an ongoing cacophony of genetic voices all speaking at once. Why is it that our bodies operate in such a chaotic way, maintaining the potential to take on characteristics of the opposite sex long after maturity? Good question, don't know. The mechanism is surely a hangover from our fishy sex-swapping ancestors, but there's no concrete answer to why it's been preserved in mammals. Parking gonads for a moment, in humans, there's no universal penis versus vagina dichotomy either. This is because a person's external genitalia develop long before their gonads do, and are controlled by completely different genes. A 5A reductase deficiency causes XY males with testes to develop something on a continuum between a vagina and a penis. Testicular feminization syndrome arises from either no or too few or ineffective testosterone receptors, which result in XY males with testes producing genitalia on a continuum from classic vagina to something more ambiguous. But seriously, I hear some listeners protest, just how common are these cases? People with non-conforming genitalia are actually more common than people with IQs over 140. That's about 1 to 2% of the human population. Lots of people. Assuming all your genes are functioning correctly, whatever that means, it's actually the hormones produced by one's gonads that make us look and act more or less male or female. Every one of us has both estrogen and testosterone acting within their bodies all the time, in various concentrations, constantly altering our behaviour. And there are several types of testosterone as well, not just one. Dihydrous testosterone grants external male genitalia during development, while another kind is converted into estrogen, which grants female typical external anatomy, but male-associated brain structures, which we'll get into in a bit. Female embryos actually have to build special proteins that bind to estrogen before it can get to the brain, if they're to preserve normal female brain development. It's bonkers. And more layers of complexity arise when we realise that every hormone needs a corresponding receptor for its effect to be felt. For example, androgen receptors allow the male hormone testosterone to bestow its effect on development. Yet all the genes for building these receptors are located on the X chromosome. This female chromosome builds the receptors for the male hormone. All hormones do is bind to receptors on cells, causing those cells to do things in response. The production of these receptors is controlled by yet more convoluted genetic pathways. Individuals with androgen insensitivity syndrome may have no testosterone receptors and present as pristine females, but are genetically males. They can have XY sex chromosomes and heaps of testosterone, but they look, act, and feel like females. There are cases of people who have lived out their entire lives not knowing this until it's finally discovered during old age. Hopefully, by now, you share my discomfort in labelling genes, hormones, and chromosomes as male or female. Attempting to maintain such trans-exclusionary nomenclature is getting quite tedious. In reality, even concepts of specific male and female behaviours becomes rather tenuous with so many conflicting variables at play in every person. Adding the intricacies of human culture and sociology to the mix gives us plenty of incentive to retire old-fashioned either-or male-female stereotypes. These are, at best, silly, in my opinion. There are enough variables on the sex chromosomes alone to make a female masculine enough and a male feminine enough to pass as the opposite gender in both social and biological contexts. This is because behaviour is controlled by the brain, and possessing an X or Y chromosome is neither sufficient nor necessary to establish your sexual identity. No matter what genes you have, multiple downstream effects are working their magic to determine how you think, act, and feel. And the cumulative effect of various brain regions may make a person feel other than what their chromosomes, gonads, or genitals suggests they should. So the best thing to do is just ask someone how they would like to be identified and take their word for it. All humans show enormous variation in anatomy and development, from how and where freckles form to the shape and position of whole organs. All of these instances are not defects, not deformities, not mutations, not anomalies, they are variations. Variations is the correct term because they are very common and generally cause no harm. Sex, sexuality, and gender identity are all subject to the same spectrum of variations between individuals and occur in every primate species ever studied. We've seen that there is no universal XX versus XY dichotomy to be found between human sexes, and no universal ovary versus testes dichotomy out there either. Because there is no standard template for male versus female development in biology, it is completely impossible to reduce sex to a binary distinction. Just like old and young, hot and cold, early and late, we should only use terms like male and female out of normal descriptive convenience. Just like biological sex, sociosexual behaviour is almost infinitely variable as well. Some animals, like cuttlefish, have 2 sexes and 3 genders. Some, such as snails, have 1 sex and no genders. Male bats lactate, and daddy seahorses give birth. Some male penguins tend to the eggs and protect their chicks. Other birds share all domestic tasks equally. There are female frogs who deposit their eggs inside male frogs. Walruses are patriarchal, lemurs are matriarchal, and our close cousins the bonobos are scandalously egalitarian. Human beings are not special in any of these respects, except that we can communicate in great detail how it feels to have a gender. Listening to people reveals that a person's gender cannot be reduced to or predicted by their biological sex. In other words, it doesn't matter what you think their sex is. What matters is who they feel they are and how they present themselves to the world. As discussed, it's perfectly acceptable for this to change as a person develops, ages, and discovers more about themselves. Gender is fluid. Just like being married, Buddhist, conservative, rich, healthy, or old-fashioned, gender is also a social construct. The term refers to a traditionally accepted way of behaving. This is always subjective, specific to a particular time, place, and society. Considering who gets to wear dresses, fix machines, bake cakes, hunt food, decorate their bodies, and lead a religious ritual, shows us that gender roles can and do change arbitrarily between cultures and time periods. For probably thousands of years, Southeast Asian cultures have recognized a third gender for people they consider to be in possession of both a male and a female soul. Shout out to my Katoi friends in Thailand. This is similar to the twin spirit people of Native America, who acknowledge a third and fourth gender. Hadra people in India, as well as some Hawaiian, South American, and African cultures, also currently recognize more than two genders. Because the words masculine and feminine don't mean anything concrete in biological terms, gender simply refers to how an individual expresses their sexual identity in a social context. This makes someone's gender a reflection of who they wish to be, given the background expectations of a particular culture. So what's going on in our heads to create all this delicious diversity? There are at least 3 areas of the human brain that are independently sexually dimorphic. I won't trouble you with yet more technical acronyms. Oh, okay, they're called BSTC, SDNPOA, and VIPSCN. Get Googling. The development of each of these brain regions exists on a spectrum, just like everything else. But even if each of these variable brain bits were just a simple on-off switch, that would still be 8 different ways that a brain could develop between basic male and basic female. Factoring in both gonad sets means that we can realistically double this. But then, as we've seen, someone could have neither or both types. This would mean that humans boast more than 16 independent configurations that could realistically be called their gender, each arising in different proportions of the population. But wait, we also need to factor in sexual orientation. A person could be any of these 16 plus flavors and also be gay, straight, bisexual, asexual, pansexual, etc. And any of these will present some conforming set of external genitalia or not. As all these facets of humans are independently controlled by separate ensembles of genes, they all present as independent variables. It's spectrums all the way down. Anyone who still wants to insist that people all fit into exactly one of 2 possible boxes must take on the daunting task of explaining away hundreds of commonly occurring exceptions, all over the globe. Instead, it's much easier and more scientifically accurate and more intellectually honest to accept that traits like sex, gender, and sexual orientation exist on a fantastically broad spectrum. We should celebrate this diversity of human nature, not stubbornly insist that because someone has XY chromosomes, that means they're a boy, and boys should act in specific ways. Hopefully, I've been able to convince you that assigning appropriate behaviours or aesthetics based on gender is on very shaky ground. Such dogmatism is not only profoundly naive, but inaccurate, bigoted, cruel, oppressive, and often tragic. To really drive this home, the Trevor Project reports that suicide is the 3rd leading cause of death for people between the ages of 15 to 24. On top of this, LGBTQ+ folk in this age range are 4 times more likely to attempt suicide than their peers. Those who experience discrimination and non-supportive families are 12 times more likely again to attempt suicide than the LGBTQ+ youth who are already at much higher risk than the general population. The harm done by social stigma born out of ignorance is truly awful. It's oh so very human of us to assign tidy, discontinuous labels to things on spectrums. Biologists need to assign overly simplistic labels to things to make the chaos of nature more comprehensible. But sex and gender are not simple concepts. Far from it. There's a huge middle ground between male and female that relies on fluctuating hormone levels and the arbitrary nature of genetic traits. Our appreciation of this, as individuals or as a society, doesn't change the scientific facts. The buffet of modern labels that we've invented to define ourselves and each other doesn't reflect anything new in our genes. If gender diverse and LGBTQ+ individuals exist in 2024, which they do, then they've always existed, at every point in human history. This variation is unavoidable, not abominable, a natural upshot of any social species that follows such a dynamic, complex, haphazard, and utterly beautiful biological recipe to concoct its sexes. This episode is a little longer and a little later than usual because of the amount of research it took. A labour of love because the topic is really important to me. We're going to look at yet more variables that affect our personalities next episode as we take a dive into the microbiome. Are all those little microbes passengers or pilots?